The effectiveness of a drug is dependent on its ability to bind to proteins in the body. The interactions that occur on the nanoscale during binding are important when it comes to understanding what changes need to be made to create a more effective medicine. Proteins are life. Mutations in DNA result in changes in the sequence of amino acids of a protein, which leads to changes in the way a protein functions and is the molecular cause of illness and aging. When this DNA strand breaks, repair enzymes fill in the gap with new nucleotides, making one strand longer than the other. When they split and replicate, one of the resulting cells will carry that mutation and continue to multiply. Targeting what causes these mutations on the nanoscale can prevent diseases from developing at their earliest stages and drastically reduce aging. The number of possible proteins in the world is approximately 2, with 400 zeros after it. Right now, we only know of around 29,000 proteins. Proteins are made up of different combinations of amino acids. These amino acids are strung together in a chain and are then folded into the proper shape. These shapes are crucial for the proteins to function. The main objective behind the folding process is to minimize the number of hydrophobic parts that are exposed to water.
The easiest expression of hydrophobicity is that oil and water don't mix.
When mixed together, oil and water will separate because oil is extremely hydrophobic. This is because oil is a nonpolar molecule and water is a polar molecule.
So the main idea is that water cannot interact favorably with this molecule, and so the molecule doesn't want to get into water.
In this example, the billiard ball on the right has a hydrophobic coating, causing it to resist interaction with the H2O, while the ball on the left has a water-loving hydrophilic coating. Notice how the impact with water affects each of the balls. That the water doesn't immediately encompass the ball on the right after impact shows how a hydrophobic molecule would interact with a wet surface. The most hydrophobic surface found in nature is the lotus leaf. The contact angle formed with water is 180 degrees, causing the water to form a near-perfect sphere.
And so you have a molecule that's sort of complicated, right? And then what people think is that when they interact with each other, there might be another molecule, right? And it talks to that molecule, it recognizes, it binds, how drugs bind to probably proteins, right? And so when two proteins interact with each other, hydrophobicity also plays an important role.
When proteins interact with other molecules, the hydrophobic properties of their surfaces influence if and where those molecules bind. In drug applications, there are millions of molecules to choose from, so determining which molecules will interact with the designated proteins, as well as how they will act, is of great importance.
So the question is then, how do we know that this surface is hydrophobic at the nanoscale? A science has developed where we try to model reality, mimic reality on a computer. You can create a movie of the system at the molecular level. You can actually watch how water molecules do their dance at the interface or at the surface of a protein or in a salt solution or whatever you're interested in. So imagine you've got a wobbly, complicated protein surface. You do this kind of calculation near it, and then you now know this is more hydrophobic, this is hydrophilic, this is intermediate, and so on. And then you can select different colors to indicate different levels of hydrophobicity or philicity. And so you can now then spray paint like the surface of a protein. And then you can look at it and say, this part must be sticky, it must go bind to this part, and so on. And so you can then start understanding perhaps or developing methods to understand how proteins talk to each other.
Using computer simulations to create particle maps allows researchers to identify common characteristics between particles.
The other part is, again, I think understanding hydrophobicity might help is in protein design. So I don't know if you've heard about protein design.
The technology exists to synthesize proteins, but the possibilities are virtually endless, so we can't try them all. By studying how proteins fold, along with how hydrophobicity affects the process, we can figure out how to create the proteins that we can best utilize without simply trial and error. Our bodies consist of about 60 to 70% water. Therefore, the properties of water are important to understand in order to know how we work. The understanding of hydrophobicity is key because it relates to interactions between water and other particles.
So you might have HIV protease, a protein that is helping the HIV virus to replicate, let's say. So you want to stop it. You figure out the structure of the protein, you figure out the active site, and then you design a drug molecule that will bind specifically to that site. What is challenging when this happens is we don't know what sort of role water molecules play in this. Right? So you can already tell that if the active site kind of looks like this, then my complementary molecule have to have the kind of curvature. So lock and key fit has to be right. They have to sort of have the right interactions maybe. But in all of this stuff, I think we still don't quite fully understand the role of water molecules. And so this is where, the research that we're doing sort of will play a role.
This research could lead to the development of specialized drugs engineered to conform to a specific disease, such as cancer, and destroy it without harming healthy parts of the body.
It's possible that some of these proteins that we haven't explored, nature hasn't explored, hold the answer. Right? And so in design of new molecules that are proteins, but they do different functions and at different thermodynamic conditions, there's a possibility of making those kinds of molecules, and then understanding of the water hydrophobicity will be at the center of it.